We've talked about speciations, and particularly with the biological species concept and thinking about isolation being the key. So the last topic I'd like to cover is this process of speciation, ways that we can classify that based on what that separation actually is. So the first would be allopatric speciation. Of course, this is the one that we think of most frequently because we actually put a geographic barrier in between these two populations, effectively stopping gene flow. We can think about two ways that this could happen. One could be a vicariance model where we might have a relatively large population and it is split into more or less equally sized populations. So think about as the continents are drifting apart, or you have the Isthmus of Panama starting to form, you may have had continuous populations, but now a barrier has been put in place, essentially pinching part of that population off from the other one. The other model we can think of is one that we've already discussed really to a certain extent when we thought about dispersal and the effects of dispersal on population differentiation when we talked about FST values. So this peripheral isolate model really is based on the fact that a population might be in a mainland population, hop over to another island, hop to another island. Sometimes it can hop back to that island. So looking at these measures of diversity can start to see what was the order of events in which this happened. One like this is particularly important when we think about population bottlenecks or founders effects, because again, the individuals that move to this new population probably are not going to be representative of the entire gene pool in the original large population. And that brings us back to this idea of population divergence then. So now we have two parts of a population that were one or even more, in some cases might be more than one population or more than two populations, and we can think about drift being one of the major causes, but we could also imagine that, well, when they get to these new areas, the environment is going to be different. The selective pressures are going to be much different. So we would expect different traits to be favored in one habitat versus the other. So even if these populations were to come back together, we'd have already had kind of this reinforced isolating mechanism in place. So geography may have been the first one, but we can put additional mechanisms in place. So that's what we can think of when we think about this idea of reinforcement. So we have populations become established. We might have post-zygotic reproductive isolating mechanisms in place. And if we have these post-zygotic ones in place, then that can even further reinforce the idea that we're going to have pre-zygotic. So if you become essentially more selective about who your mate is, then you'll end up having greater success, because if you have a mate that is similar to yourself, then your offspring are going to have more success. If you find a mate that is not like yourself, then your hybrids are not going to be very good. So it's kind of nice if you can say, well, I'm going to stop this before it even gets started, as the case with the phlox populations in Texas, that they have the color differentiation between them. And this color differentiation means that they're going to attract different pollinators, and by attracting different pollinators, they avoid cross-pollinating with the other species, and they avoid having lower fitness. But of course, all this can be a bit leaky, right? So when we talk about this speciation continuum, most of the time we don't have a strict time point or a strict solid geographic barrier. So there's always going to be some migration, there's some gene flow that can go across there. So we talked about this when we just talked about separation of populations and dispersal of populations. So how wide that zone is, how rapid the alleles change within that zone gives you an idea of how strong of a selective pressure there might be and how much migration there is. Because again, if we increase the migration, we're going to have a wider cline or a wider hybrid zone. If we have really short migration distances, we'll have a narrower hybrid zone. Or if we have strong selection, we're going to have a narrower hybrid zone. Occasionally we can think about cases where the hybrids actually are stable, and these hybrid populations can persist through time. And that can be the case where you can have two of the parental species can remain separate, but the hybrids are just continually reproduced. So they don't necessarily have a disadvantage or an advantage, they're just very, very commonly made. And we see this a lot with a lot of plants where we call what's called a hybrid swarm because these two parent species readily reproduce and they always have a new influx of hybrid offspring. But we can also have cases where the hybrids actually do have an advantage and they do better in maybe a novel habitat that neither one of the parent populations live in. Then we'll go to our two cases of speciation. Again, this is looking at speciation through space, but now we'll look at ones where they're in closer contact. So there's not a hard geographic barrier. So parapatric speciation, they're butted right up against each other. Sympatric speciation, it is actually happening within a population. So we'll talk about sympatric speciation first. And again, I'm going to talk about these very, very generally. I don't want to get too much into the details on this before the exam. We can just think about sympatric speciation having variation within a population. So we can have polymorphism. So we have different types within a population and they're stable. We see them frequently, but then we have assortative mating. So you're selecting your mate based on what they look like. So the example that they give within the textbook is looking at the cichlids in Africa and how these fish populations are within the same general area, yet they're starting to have assortative mating. Parapatric speciation is really kind of like what we talked about again when we think about migration and selection or when we think about hybrid zones. So we'll have some sort of decline and then we'll have this border between them depending on how much gene flow there is. We might see alleles going one way or the other, or we might really see distinct sets of alleles in both cases. So in this slide we see two species of lizards. The first one, the Holbrookia, is one where they have a very strong selection, where they have primarily light-colored alleles, so each bar is an individual lizard in each one of these panels. And the color of the bar shows the percentage of the genome that is one morph or the other. So on the left, we have ones that tend to have a light more of the light-colored morph alleles associated with it. On the right, you have more of the dark-colored alleles. In the bottom two panels, we have the same sort of a pattern, but either migration is greater between these species compared to the Holbrookia, or the selective pressures aren't as strong. Again, so emphasizing this idea of strength of selection. If selection is really, really strong, we can negate any homogenizing effect of migration. So in each one of these graphs, we can see changes of the strength of selection. So we can see the relative fitness of the migrants, the relative fitness of the hybrids. In panel A, they're both really, really close to 1. So they're not much different than what the resonant fitness would be. So the resonant fitness in all of these cases would be 1. As we go through each one of the panels, we have stronger and stronger selection against migrants and against the hybrids, which are essentially intermediate between the 1 and the migrants. So if the resonant fitness is 1, migrants is 0.8, hybrid is 0.9, etc. So when we think about these really strong selective pressures, we start to see a really, really strong divergence, even if we have a lot of migration. And again, we can just think about this idea of reinforcement with something like ecological speciation. So we might have this geographic border in place, but then we're going to start to actually have new sets of parameters that take place. And we're going to start to see flowers that are going to be living in one elevation. Pollinators tend to be in one elevation versus another. So they're going to start to be selected for different types of traits based on who their pollinators are and what's present in that new population in the first place. Okay, so this brings up the idea of, can we see parts of a genome that are particularly targets for speciation? And this is the exact same thing that we saw when we looked at our paper on the lizards, the urban and woodland lizards in Puerto Rico. So again, if we think about the FST values as our relative measure of diversity within populations versus between populations, high FST values mean that you have a high level of divergence. Low FST values means you have a low level of divergence between them. So we know that if we increase heterozygosity, then we're going to be increasing at least the potential amount of divergence there is. So the more heterozygosity there is, the higher that FST has to be to show you that it's essentially beyond what you would expect from chance. So these outlier loci are the ones we would focus on for ones that we think are going to be strongly selected for. So again, in the lizard paper, they did that genome-wide scan to see what were the loci that seemed to be the target of selection. This is what we see here. So this is what we might see in a population of mosquitoes. So we can say along individual chromosomes, we can look and see, are there values that go higher than what we would expect by chance for divergence on these populations? And again, this is what we saw in case of the width of the toe pad or the length of the toe pad, or in some cases, the different immune responses that we saw in the different lizards. But we can think about kind of a little island of divergence. So there's either a locus that's going to be selected for or selected against. And again, because of linkage, we might have regions just before and just after that that are part of this island of divergence. So there's a specific location where we see the signature of one population or species versus the other one. Now, the very last topic I'd like to cover is thinking about the idea of biodiversity. So we've really kind of focused everything on speciation, taking one lineage of one population and making one species into two. But we can really think about what happens as we kind of look from kind of a higher view, like the higher view of the Earth and a higher view of time, and start to see what about larger patterns of biodiversity. And we can see that through time, speciation rates have changed, or divergence rates have changed. Sometimes we might have a relatively even rate of speciation in some groups. Some groups may have actually diversified very early on in the history of the Earth. Some of them may have actually diversified much, much later. So thinking about these individual patterns is what I'd like to think of for kind of this last topic within our speciation and diversification unit. So as we think about the history of the Earth and we look at the history of the Earth, we can see that over time we have had an overall increase in the number of families and an increase in the amount of diversity. But we've also seen that we do have kind of periods of extinction that go on through this time. So when we look at this a little bit more closely, we can see overall, our overall extinction rate actually has gone down through time because we've had more and more species present through time. But we do have these red dots that represent these 5 mass extinctions that have been recorded in the fossil record. And this is what we want to focus on when we think about big crashes in diversity, and then how can we recover that biodiversity. So if we think about each one of these mass extinctions, on the x-axis of this, we can look and see what our relative diversity is. So our relative diversity, we can have that averaged out as 0, and then we can have a relatively low biodiversity, relatively high biodiversity. So right after a mass extinction, we're going to have a relatively low overall diversity. And you can see on the upper left-hand corner of this graph, when we have a low overall diversity, each one of these labeled dots is right after a mass extinction. So these higher rates of diversification happen right after each one of these mass extinctions. So we can think about these globally. So from a standpoint of biogeography, where do we find different clades as they're spread throughout the world, and how are they related to each other? We can really kind of think of what are 2 of the big drivers that seem to affect how fast diversification is going to occur. So one of them is just how long has it been since divergence. So if we've had something around for a long period of time, there's just more time for it to happen. And then we can also think about, similar to what we can think of for carrying capacity, of how many individuals of a single species can be sustained in a habitat. We can also think about how many species a region can actually sustain. So in graph A at the bottom, we can compare a temperate and a tropical. And tropical environments are much more productive. They have much more food available, so they can sustain a higher wealth of species than what a temperate one can. And we also tend to see relatively higher diversification rates. In the tropics, we have a lot more specialized relationships, a lot more mutualisms. These mutualisms usually lead to coevolution and greater diversification rates. In panel C on the far right, we can just think about the tropics compared to the temperate zones. And the tropics, generally, the land itself is older than the temperate zones, especially if we think about as the glaciers retreated, a lot of the land in North America was not necessarily ice-free. So species had to migrate back up north before they could even begin to diversify. But the tropics have been, at least for the last 1 million years or so, have been relatively stable, so there's been lots of time for them to maintain a large diversity. So again, this brings up the idea of how do we start? So tying this idea with mass extinctions and seeing this explosion of biodiversity, we think about this as what we call adaptive radiation. So we have a relatively small number of taxa, a relatively small number of clades that have survived this event, and that's where we have to start with, right? So when we talk about constraints of natural selection, every tetrapod is built like a tetrapod, right? So we think about our arm bones and our leg bones, and they're the same for all tetrapods because that's what our original tetrapod body plan started with. So now we have to think about, if we're going to become highly diverse, we have to start with, well, whoever is left there. So again, if we think about these adaptive radiations, one I brought up already of mass extinctions, but we could also think about a colonization event, right? So one small group of individuals of a population move on to an island, they've colonized it, and everything from then on is going to be developing from that. So the marsupials in Australia are a really good example. So the only mammals that are native in Australia are marsupials. So they started off with a limited number because that's what made it there and they colonized, but then it separated off and all mammals came from the basic body plan of a pouched animal. We can also think about new evolutionary innovations. So are you starting to be an herbivore? Do you have wings? Do you have other sort of behaviors? So is there a new sort of innovation that you have that nothing else has, and now selection has something new to act on? So let's talk really briefly about what we would expect to see for adaptive radiation to occur. So if this is going to occur in the first place, again, a big assumption of adaptive radiation is we're starting off from a small number of groups and becoming much more diverse. So the very first criteria is going to be common ancestry. So all the species should be derived from a recent common ancestor. So each one of these, you can see that there may have been an early origin of each one of these groups, but then through time, you saw each one of these bubbles starts to get wider and wider. So we can see a big explosion from one point. So it gets bigger and it gets bigger from that early common ancestor. We also need to think about what the traits are and what's being selected for. So whatever niche the organism is occupying, they have to have the phenotypes are going to be associated with that. So we have to have distinct phenotypes associated with that environment. So in this case, we might say, well, we have different seed mass as a trait, and different seed mass might be found in a shade-adapted species versus a light-adapted species. Then we have to think about the traits themselves. The traits themselves actually have to give you some sort of a fitness advantage. So we've already talked about this when we talked about changing adaptations and being able to recognize adaptations. So population A and population B have different morphs, and A is always going to outcompete B as long as it's in the A environment. B will always outcompete A when it's in B's home environment. And the last one is this idea of rapid speciation, because again, if we're talking about adaptive radiation, we're talking about a relatively quick shift. So through time, we can see kind of two general patterns. So one we might think of as competitive displacement, where you have clade 1 in the upper panel starts off really diverse, clade 2 originates, and then clade 1 slowly disappears. Clade 2 kind of slowly increases. So that gives us more of an indication that maybe it was more of a competitive displacement rather than just an explosion. As opposed in the bottom panel, we see clade 1 was highly diverse, then we may have had a mass extinction event, clade 1 disappears, clade 2 is around right at the beginning of that mass extinction event, and a few members survived and increased in numbers to have high levels of diversity. So to reiterate, the common criteria for adaptive radiation: 1, all the species have to come from a recent ancestor. We have to have phenotypes that are associated with a given environment. Those phenotypes actually have to have an adaptive value within, or those traits have to have an adaptive value within that environment. And we have to have rapid succession, so coming from a single ancestor and rapid speciation from that ancestor. So then the last thing to think about would be thinking about these patterns and richness and higher order, the same patterns that we can think of for biogeography, with thinking about the effect of time since diversification and diversification rate. Some clades might actually be more likely to have higher levels of diversity than others. So we can think about, are there groups that have higher mutation rates? Are there groups that have higher latent genetic variations? Remember, standing genetic variation is key with our quantitative traits. We might have the alleles to be much taller than what we are, but we don't see it in the original population, but we see it in a later population following selection. And then also you might guess that motility is going to be really, really high. So if you're going to have adaptive radiation, you need to be able to get to these new areas. So this is tied with the idea of finding new niches, finding new habitats. So this really wraps up all of our topics that we're going to cover for speciation and diversification. I really hope to be back on Wednesday. I'm not feeling terrible, but I still have a fever and my head still feels like it's pretty split open right now. So I hope to see you Wednesday. If not, then I will hold a Zoom meeting so that people can ask any questions that they have.